Experiment attempts to understand the effects of nutrient restriction on tadpoles. This was done by depriving the tadpoles of additional food in their environments. Researchers found that tadpoles that were nutrient restricted experienced slow physical growth, a volume decrease in the optic tectum, which is a structure in the dorsal area of the midbrain, and slowed brain development. It was also found that this damage could be reversed, or at least significantly improved, if food was reintroduced into the environment in 9 days or less.
Our study tested if limiting nutrient access to tadpoles in early development, when progenitor cell proliferation is normally high, affects growth of the optic tectum and tectal cell proliferation. It also tested whether or not tadpoles can survive a period without nutrients and recover tectal development with access to food after the period of deprivation.
Here is some general terminology and information for understanding our research. Frog development can be broken down into 56 stages from conception to maturation, which takes approximately 58 days. We studied these organisms when they are in stages 46 to 48, which last for about 10 days in total. These stages are particularly important developmental stages for the brain that dictate the organism's further development. The operculum, peritoneum, and cement gland are 3 visible features that, along with several others, play an important role identifying the stages of development for the tadpoles. During stages 46 to 48, physical features like these
show distinguishable changes that are good for identifying what stage the frog is currently in. The world is a constantly changing place, and organisms must learn to adapt to these changes. For example, bears hibernate in the winter to accommodate for the lack of nutrients.
The most common challenge faced by animals in nature is nutrient scarcity. Organisms have different coping techniques like hibernation or diapause.
There are 2 main responses to nutrient scarcity. Nematodes exemplify this. Constant access to nutrients is imperative to proper development. This study is important because the effects of nutrient restriction on brain development are currently unknown. This study set out to determine these effects in tadpoles. The optic tectum was the main portion of the brain studied during this experiment. The results from this study allow us to use tadpoles as a model organism for the effect of nutrient restriction on brain development. This can then be more widely applied to other organisms.
Tadpoles were bred in the lab. Each group or clutch of tadpoles came from one separate pair of breeding tadpoles. The tadpole's development was characterized by the Nequip and Faber stages of tadpole development, except stage 47, which was divided into early and late substages. In each experiment, about 250 stage 41 tadpoles were placed in a Steinberg solution without food. At stage 46, 10 tadpoles were euthanized with MS-222 anesthesia and fixed in 4% paraformaldehyde. The rest were divided into 3 groups of about 80. Group 1, the control, were fed twice a day. Group 2 were the nutrient-restricted group and had no food. Group 3 were the delayed feeding. The delayed feeding tadpoles were not fed for 3 days, but after this time they were fed twice a day. Tadpoles were fed 500 microliters or 30% slurry of Xenopus Express tadpole food. Once a day for 10 days, 6 to 11 tadpoles from each group were euthanized and fixed. The experiment was then repeated, but the delayed feeding group was not fed for 7 to 13 days. Nikon stereo microscope was used to view the fixed organisms. The initial size of each group of tadpoles was compared in order to determine if each group started out at significantly different lengths. No significant difference was found and the data could be pooled. Tadpoles underwent behavior screening for avoidance multiple times. The tadpoles that made it through all screenings were included in avoidance behavior experiments. After fixing the tadpoles, their brains were viewed under an Olympus FlowView 500 confocal microscope at 20 times magnification.
The cross-sectional area of the largest part of the tectum was measured and different kinds of cells within the tectum were counted. Many statistical tests were run, and although behavior was found to be normally distributed, cell counts in the brain were found to be statistically different.
The data collected from measuring the body lengths of fixed tadpoles is displayed in the graph. The nutrient-restricted, or NR, and the delayed feeding, or DF, groups remained statistically comparable to the continuously fed groups until day 4. At day 4, the fed groups continued to grow, while the nutrient-restricted and delayed feeding groups did not. The growth of the nutrient-restricted group continued to decline for the rest of the experiment, while the delayed feeding group began to grow faster once feeding was restored, and their growth became statistically comparable to that of the continuously fed group between days 5 and 10. Next, each tadpole was categorized by the developmental stage from which they were fixed. These developmental stages were categorized in 2 ways, first by organ development and second by morphological features. There are many conditions that contribute to a tadpole's ability to grow. Specifically, the experiment focused on the rearing conditions listed above. Stage 46 lasts for around 1 day and is defined by the criteria above. Stage 47 lasts for 2 days but is separated into an early stage and a late stage because of their distinct physiological differences. The criteria for early stage 47 are listed above. The criteria for late stage 47 is listed above. The pictures show the morphological differences between various stages of development. Stage 48 lasts for the remaining days of the experiment and is defined by the criteria above. On day 0 of the experiment, most of the tadpoles were in stage 46 with an average stage of 46.81. By day 10 of the experiment, the continuously fed group had an average stage of 47.75 with much of their group in stage 48. In contrast, the nutrient-restricted group had an average stage of 47.91 with none of their group reaching stage 48. Between days 2 and 7, the delayed food group was statistically less than that of the continuously fed group. On days 7 and 8, the group became statistically comparable to that of the continuously fed group. However, this number dropped for days 9 and 10, where it again became statistically less. The delayed food group had an average stage of 47.53, with some members of the group reaching stage 48. Avoidance behavior was tested through a range of behaviors, some of which required visibility and others did not. Throughout the experiment, there were no differences among the 3 test groups when it came to avoidance behavior. After day 8, there was no significant changes in avoidance behavior in any of the groups. This lack of variability is shown in the graphs above. To measure brain development, the optic tectum was measured across the widest part and used as a proxy for development of the midbrain. The optic tectum of the continuously fed group grew quickly after day 1, whereas the nutrient-delayed and nutrient-restricted groups had slow growth. In the nutrient-restricted groups, slow growth maintained, and this growth even declined in some instances. This is shown on the graph above. The nutrient-delayed group quickly caught up with the continuously fed group once food was reintroduced on day 3. In order to measure proliferation rates of brain cells, pH3 markers were used to label metaphase dividing cells of the brain. This then could be used to measure how quickly and often cells of the brain were dividing. The control group showed that proliferation naturally decreases during the time period that stages 46 to 48 takes place. The nutrient-restricted group showed depressed rates of cell proliferation for the entire experiment. The nutrient-delayed groups had lower rates of proliferation until food was reintroduced into the system, in which case proliferation rates increased and maintained the levels of the continuously fed group for the rest of the experiment. Cell death in the brain was measured because this can be caused by a lack of nutrients or increased proliferation. Syntax O was used to label dead cells in the optic tectum, which is used as a proxy for the rest of the brain. There were no significant differences in the number of dead cells found in the brains of any 3 test groups, as shown on the graph above. To further explore nutrient restriction, extended recovery studies were also done on nutrient-restricted groups. In one group, food was restricted for 8 days, followed by 4 days of feeding. Once feeding was reintroduced, proliferation rates increased to that comparable with the continuously fed groups. Similar results were found when a group was delayed food for 9 days and fed for 3. However, once food was delayed for 10 days and feeding resumed for 3 days, there was no recovery of proliferation in cells of the brain, despite the fact that food was found in the gut of tadpoles during recovery. This means that tadpoles were obtaining nutrients actively by eating, but could not recover to proliferation rates comparable of that of the continuously fed groups if food was delayed for 10 days. The dark picture above shows pH3 labeled cells from a representative sample of brains from each of the test groups. This shows general proliferation rates.
The graph shows that the control and the 9 or less day restricted tadpoles are statistically comparable. During the period of stasis, tectum stasis and shrinkage were observed. However, syntax O showed this shrinkage was not caused by cell death. This is contradictory to the studies done on C. elegans, which were able to maintain a state of nutrient deprivation through mutated insulin signaling systems with accompanying cell death. The tadpoles also remained active during this time period, unlike bears and many other organisms that resort to hibernation or other forms of inactivity to cope with nutrient restriction.
Tadpoles cease to grow over the 9-day nutrient restriction period, but when they are fed again, their growth and proliferation does not exceed that of the continuously fed. This suggests that proliferation is controlled by a mechanism in the optic tectum. Other animals, like certain species of fish, show similar kinds of growth arrest with similar mechanisms to that of the C. elegans. A variety of studies demonstrate the correlation between nutrient restriction and decreases in proliferation. There is also evidence that there are mechanisms in the brain that control the process of proliferation. Proliferation requires a lot of energy, which could be why it has such a strong correlation with prolonged nutrient restriction. This energy-saving technique could help organisms sustain life in extreme environmental conditions.
This study will create a better understanding of to what extent tadpoles recover from nutrient restriction. This could lead to more exploration of how different animal populations recover from this stressor, as well as long-term physiological and morphological effects of nutrient restriction. This would allow for comparison between various animal models and their nutrient restriction limits.
Some ideas for future improvement of the study are studying tadpoles after the surgical removal of yolk, using larger group sizes, and studying frogs that the tadpoles result in. This would allow us to collect additional information and determine the lasting effects of this experiment on tadpoles.